What makes nucleic acid amplification the heart of modern molecular biology? Have you ever wondered how scientists detect even the tiniest traces of genetic material in a sample? Today we'll walk you through exactly how this powerful technique works in the lab, step by step. NAAT explained, amplifying genetic information with precision. At its core, NAAT allows researchers to multiply a specific DNA or RNA sequence millions of times, making it easier to analyze. Unlike simple detection methods, NAAT uses enzymes to replicate nucleic acids exponentially. Whether it's PCR, RT-PCR, or isothermal amplification, the principle remains the same. Turn a single molecule into enough material for clear analysis. How to perform nucleic acid amplification testing, from sample to signal. Let's get practical. First, prepare your nucleic acid sample. Ensure it's pure, free of inhibitors, and properly quantified. Next, set up your reaction mix. Template DNA or RNA, primers, nucleotides, buffer, and the polymerase enzyme. Then, program your thermal cycler denaturation, annealing, and extension. Cycling through these stages replicates the target sequence exponentially. After amplification, analyze your product using agarose gel electrophoresis or fluorescence detection to confirm successful amplification. How to avoid amplification errors. Tips from experienced researchers. Contamination is really the enemy of amplification. Always make sure to separate your pre- and post-amplification areas, use filter tips, and include negative controls. It's also important to optimize primer design and reaction conditions for your specific target. From basic research to biotechnology, why NAAT is essential. NAAT isn't just a lab routine, it's a foundational technology behind gene expression studies, environmental monitoring, and advanced biotechnological innovation. Mastering this technique truly opens the door to a wide range of molecular applications. BOC Sciences, your trusted partner for nucleic acid products and services. At BOC Sciences, we support your amplification experiments with high-quality nucleotides, oligonucleotides, enzymes, and custom molecular biology services. Whether you're designing primers, optimizing amplification workflows, or scaling up for industrial R&D, our experts are here to help. Visit our website to explore our full range of nucleic acid solutions and accelerate your research today. If you found this tutorial helpful, don't forget to like, subscribe, and stay tuned for more biotech insights from BOC Sciences.